tanto vamos dizer, de base laboratorial quanto as de base tecnológica, né, de tecnologia da informação. Então, a gente tem hoje aí uma série de palestras, né, onde que a gente vai discutir um pouco, nossos convidados e convidadas vão...
Oi, Alan, me ajuda aqui, querido.
E em seguida teremos, né, no final do nosso trabalho aqui, uma mesa redonda para a gente fazer um apanhado do que a gente for discutir, né? Então, me apresentando, sou Leonardo Teixeira, sou um dos coordenadores aqui no Instituto Senai de Inovação em Biosintéticos e Fibras do Senai Cetiqt. Esse é um evento que, né, estamos co-realizando junto com o Chemical Abstracts, né, da American Chemical Society, a rede MCTI Embrapi de Inovação em Bioeconomia, né, e o próprio Senai. Então, passando um pouquinho pela nossa agenda de hoje, além dessa breve abertura, né, teremos agora em seguida uma palestra da professora Vanderlan Bolzani, né, que falará sobre a prospecção de moléculas da biodiversidade brasileira e produtos de alto valor agregado. Em seguida, teremos uma palestra do Brian Harkelroad, né, que ele é diretor de desenvolvimento de soluções do CAIS, onde vai falar um pouco dessas ferramentas que o CAIS oferece para essa prospecção e a importância para o mapeamento da biodiversidade brasileira. Em seguida, teremos o professor Reginaldo Luceno, falando um pouco sobre a iniciativa de mapeamento de diferentes bancos de dados de moléculas existentes. Teremos uma breve pausa para um coffee break às 2h30, né, voltando às 3h30. Em seguida, a nossa pesquisadora aqui no Instituto Senai de Inovação, a Yamin Esquizani, vai falar sobre o Biodiversity Business Discovery, que é uma iniciativa que o nosso instituto está lançando, que é exatamente voltada para a identificação de oportunidades de negócio na biodiversidade. E ali, perto das 4 horas, entre as 4 e 5, teremos uma mesa redonda com todos os convidados, né, tanto os presenciais aqui quanto os que estão online, para a gente fazer a discussão do que a gente comentar hoje, né, seguido de um breve encerramento. Então, eu queria aproveitar e já passar de novo a palavra para a professora Vanderlan Bolzani, né, que fará a sua apresentação. Professora, fica à vontade, se quiser se apresentar também em mais detalhes, né, a palavra é sua.
Pode falar que já estou te ouvindo.
Olá, uma boa tarde para todos. Eu gostaria agora de colocar os slides para começar a falar. Antes de agradecer, ele está me ajudando, porque a gente não está conseguindo colocar. Só um segundo.
Não tem problema, professora, a gente aguarda.
Por quê? Tá vendo? Aqui, ó,
Eles estão te vendo?
Vocês estão me vendo, né?
Sim, estamos sim.
Então, é que eu tô tentando aqui o professor Ian Castro, que é um...
Onde que está a sua apresentação?
Essa aqui, que
Essa
já
aqui?
está aberta.
Tá bom, vamos lá.
Já tinha aberto, ó,
Só
tá
um
vendo
segundo.
esse aqui?
Vamos ver se eles conseguem ver. Pergunta para a plateia.
Vocês estão conseguindo ver?
Agora sim.
Ah,
Ok.
então tá
Então
ótimo.
começa no número 1.
Então vou começando.
E aí, você
Então só vou aqui.
Isso.
Muito obrigada, Ian.
Imagina.
Vocês desculpem-me, porque eu estava marcado na minha agenda 13h30. Só agora eu tenho que correr. Eu gostaria de agradecer imensamente o convite que me foi formulado para participar desse evento tão importante, principalmente agradecer ao Paulo Coutinho e a toda a equipe que está aqui presente, principalmente o Leonardo Teixeira, que fez a abertura, e todos os demais que vão falar, inclusive o Brian. Muito obrigada. E, na realidade, puseram aqui um título que eu vou tentar explicar sobre a importância dos produtos naturais, já que eu sou um especialista na área, venho trabalhando há tanto tempo. E o que é que eu decidi fazer? Eu coloquei aqui um breve sumário do que a importância da biodiversidade, depois por que trabalhar com a biodiversidade do Brasil é importante e por que temos tão poucas coisas, e terminar naquilo que é o sonho da professora Vanderlan, que é deixar uma base de dados de produtos naturais certificadas para o país. Logicamente, todos aqui sabem do valor da biodiversidade, mas no momento atual que nós estamos aqui tratando com o Senai de produtos da biodiversidade de valor agregado, a biodiversidade extrapola o valor que ela pode trazer para a espécie humana em sentidos econômicos. No momento atual, onde nós presenciamos tantas catástrofes, elas são decorrentes da falta de uma política de Estado que proteja os nossos ambientes, principalmente as nossas florestas equatoriais, que são únicas. Então, esse é um dado extremamente importante. Além de trazer benefícios para a espécie humana, como medicamentos, suplementos alimentares, fragrância, etc., etc., eu acho que muito mais importante é regular o clima, é proteger o solo, fazer ciclização de nutrientes, e isso é extremamente importante para a nossa existência. Uma coisa fascinante também nos produtos naturais, quando nós fazemos uma linha do tempo, é verificar que alguns produtos da natureza mudaram a história da humanidade através dos séculos. Essa linha do tempo mostra muito bem algumas substâncias que até hoje marcam a presença, independente delas terem sido modelos para a síntese orgânica. Um exemplo muito comum, que foi muito utilizado agora com a história da Covid, que é morfina, porque continua sendo ainda um princípio ativo para dores terminais. E por mais que se fale da síntese em 5 etapas que foi feito pelo Hudley nos Estados Unidos, morfina ainda é utilizada nas grandes plantações que existem no Afeganistão e de onde se tira essa substância. Logicamente que com os avanços, né, da ciência de plantas e principalmente para a medicina, eu gostaria de destacar aqui um dado importante, que mesmo que nós tivemos uma diminuição dos investimentos e principalmente do mercado bilionário, que é o mercado de fármacos no mundo, vocês podem perceber que ainda nós temos muito que ganhar se nós tivermos a inteligência, e muito mais do que isso, uma política de Estado voltada para bioprodutos. Temos uma biodiversidade fantástica, como todos vocês sabem, não precisa nem falar dos biomas que nós temos, principalmente aqueles genuinamente brasileiros, como é o caso do Cerrado e da Caatinga, que tem uma composição molecular muito especial. Essa visão do mercado farmacêutico brasileiro também é muito interessante, como vocês podem ver, os dados colhidos em 2022, eu acho que é extremamente importante, mostrando que mesmo com essa diminuição, nós temos ainda um mercado muito valioso no que se refere ao mercado de fármacos, e, ultimamente, eu tenho levado em conta saber sobre os registros de patente de principalmente de fitomedicamentos, não, porque nós temos fitoterápicos que são misturas padronizadas, né? Nós temos um dado importante também aqui para discutir no Senai, que eu acho que isso é de interesse de vocês. Na minha concepção, patente é um bem industrial. Por que os pesquisadores têm que ter essa ambição de ter patente? Patente custa. Patente só tem valor quando tem um setor empresarial ou um setor de Estado que assuma. Então, aqui nós vemos muitas patentes em nomes pessoais, e isso é um processo que, no país, tem muito a ver quando nós estamos tratando da biodiversidade de produtos de valor agregado. Pode ver, a maioria tem Unicamp, aí tem o nome de várias pessoas, tem Botica Comercial, tem a Natura, mas muitas patentes individuais e de universidades. A comercialização também de medicamentos no país, é isso que eu gostaria de chamar a atenção para esse gráfico recente que eu coletei da Anvisa. Vejam que o mercado de fitoterápico, nesses últimos tempos, é um mercado extremamente importante no país, né? E olhem as empresas que respondem por estas patentes, principalmente. A empresa que mais tem patentes e é a maior empresa brasileira, mas da América do Sul, é o Laboratório Axé, seguido por uma série de outros laboratórios aqui, que vocês podem ver, não vamos entrar em detalhe para eu não perder o meu tempo, que já passou. E quando nós olhamos com relação aos produtos naturais, todos vocês conhecem, isso aqui foi um trabalho feito pelo Laboratório Axé, com a participação do Calixto, nós temos uma mistura que nós jogávamos no lixo quando eu fazia mestrado e doutorado de alfa-muleno e transcariofileno, que são uma mistura que foi isolada dessa planta, que é Cordia verbenácea, que é o princípio ativo do axé-flam. Então, quantos produtos poderiam sair da nossa biodiversidade? Mas, recentemente também, outro dado de 2010, é um produto que todo mundo conhece, passiflora incarnata, e que virou um medicamento, e o da ápice fitoscar, que é uma planta do cerrado que nós trabalhamos muito no passado. Então, dentro desse aspecto, nosso laboratório, que quando eu comecei lá em 98, cheguei aqui ainda muito jovem, com mestrado e doutoramento com Otto Gottlieb, um grande cientista na área de produtos naturais, entre os 4 prêmios Nobel daqui do Brasil, que foram indicados por um prêmio Nobel americano, tinha esse olhar para a natureza, mas ele não estava preocupado em isolar produtos para o setor empresarial para curar ou para melhorar a vida das pessoas. Ele queria conhecer a natureza, que é o princípio básico para se chegar aonde nós queremos. Montamos um laboratório de ponta, altamente especializado, foram muitos investimentos via FAPESP ao longo desse tempo, inclusive alguns FINEP. E só para mostrar aqui algumas substâncias que nós isolamos, que tinha tanto valor, porque um desses produtos aqui é um princípio ativo da Christian Dior, foi trabalhado no nosso laboratório, e nós fizemos isso com parceria com alguns laboratórios, que é o caso da Calicofilo, que tinha interesse no mulateiro, e era uma empresa nacional. Aqui, novamente, um trabalho que nós fizemos é um trabalho de doutorado acadêmico, mas fizemos parceria também com a Eurofarma, que é uma grande empresa nacional, que naquele momento, a substância que nós isolamos era muito interessante, que é a tubirnatina, que tinha esse dado aqui de inibição, quando comparado com galantamina, e não tínhamos como produzir isso em grande quantidade para ir adiante. Este é um outro problema da química dos produtos naturais, porque a natureza ela produz em miligramas, porque ela não produziu absolutamente nada para nós, mas para a sua regulação e sua adaptação, e assim sendo, nós temos que olhar e imitar, como fez a maioria dos países desenvolvidos. Fazem a síntese, e a síntese depois, se tiver passar por todo o crivo de um medicamento, vai para a fase de produção. Novamente, desse trabalho, o que sobrou foi uma coisa muito bonita do ponto de vista científico. O professor Zenck, que é responsável pela biossíntese de todos os alcaloides indólicos já isolados no mundo e que tem biossíntese comprovada, porque é muito importante saber a origem de como se forma. Ele ficou encantado com esse trabalho, porque nós isolamos a turbinatina, que é um indólico, e a natureza é belíssima, ela glicosila essa posição aqui, que é inédita, não existia, e a comprovação disso numa planta da Amazônia serviu para confirmar essa via metabólica desses alcaloides extremamente complexos. Um dado extremamente bonito do ponto de vista acadêmico foi essa conquista. Outra coisa também que nós fizemos no passado, que foi interessante, trabalhar também com espécies de farbácea. Esse trabalho rendeu uma patente na Natura, foi premiado no 3º lugar no Prêmio de Inovação do Natura Campus, que todos vocês conhecem, com uma composição química muito, muito espetacular, principalmente porque são aromáticos, e aromáticos simples são mais fáceis de serem sintetizados. Diante desse panorama, como eu falei para vocês, será que patente para mim é um bem industrial? Mas nós fizemos patentes. Aqui tem algumas delas, que foram feitas, a que foi feita com cássia turbinatina, que nós chegamos a fazer fase pré-clínica 1 e 2 com a ápice, mas abandonou porque nós não tínhamos condição de produzir a quantidade imensa, e a síntese não é uma tarefa simples, e nós não somos químicos sintéticos, mas no contexto nós temos todas essas patentes, e não sei se isso é um dado importante ou não, porque patente, para mim, é um bem industrial. E, diante de tudo isso, o que, só para entrar no final dessa apresentação, que é a base de dados, que eu acho que interessa tanto ao Senai, principalmente para nós que agora investimos muito, eu gosto da frase do Joly para sintetizar tudo o que eu falei. Na realidade, é uma coisa fascinante trabalhar com a biodiversidade, mas nós podemos ter oceanos de dados, rios de informação, riachos de conhecimento, gotas de entendimentos. Então, como fazer uso sustentável da biodiversidade? Então, aí é como surgiu a forma que eu imaginei, depois de tanto trabalho feito, por que não organizar isso numa base de dados? Por que não certificar tudo que nós fizemos? Esse foi um trabalho de uma aluna de doutorado, lá no passado, quando fez a tese. Esse trabalho foi muito importante, fez um impacto internacional. Em 2017, voltamos a fazer o trabalho também falando da base de dados, muito importante. Outro dado que vale a pena citar, que na página do ICBG, que é uma página extremamente importante, aparece novamente o nosso dado aqui, numa revista com índice de impacto 45, novamente com base de dados de milhões de substâncias, a NUB aparece novamente. Tudo isso levou a... E, em 2016, numa reunião no Ministério de Ciência e Tecnologia, em Brasília, fazer uma apresentação, porque eles me convidaram, que tinham interesse em investir para que o Brasil tivesse uma base de dados de produtos naturais. Fomos, tivemos conversa e não resultou em nada. Isso foi publicado no mundo inteiro, e aí tem uma linha do tempo muito rápida, porque eu já falei aí sobre a base de dados, apoiada inteiramente por um INCT, que eu sou coordenadora, que é um projeto nacional no Brasil, mas a verba FAPESP substancial, com mais 2 projetos da base com a Alemanha aprovados, e com isso nós fizemos toda essa linha do tempo e fizemos um contrato de serviço para adquirir todas as substâncias que já tinham sido isoladas da biodiversidade brasileira até 2015. Isso teve um custo de $100 mil, que a FAPESP apostou e apoiou, e hoje nós temos essa parceria, e isso é um desafio enorme, porque não basta apenas o CAS liberar as 55 mil substâncias. Nós temos que ter profissionais na área de informática, e principalmente nessa área de transição de como administrar esses dados, para colocar dentro dessa base de dados, e assim atender a todos o que nós imaginamos que a composição química, junto com toda a informação botânica, taxonômica, de princípios ativos, de farmacologia e até adiante da farmacologia, possamos ter para tudo que já foi isolado da base do Brasil. Nós temos esses dados, são 32.524 artigos publicados. Isso é uma tarefa insana quando compara, porque não é feito automatizado, nem aqui, nem nos Estados Unidos. É uma tarefa que faz milhares de pessoas. Mas nós estamos muito esperançosos de que agora possamos nos organizar e ter toda essa informação. E por quê? Porque depois que esse trabalho que saiu recente é muito interessante, é de um colega nosso, quando nós colocamos a base de dados comparada, as substâncias que já foram isoladas aqui, elas têm uma aproximação muito maior com relação à estrutura do que está no comércio de fármacos no mundo, principalmente com relação às substâncias mais sofisticadas do ponto de vista de esqueletos. Isso é extremamente importante, porque é um dado a mais para a importância das substâncias que saíram dos ambientes equatoriais e tropicais do Brasil. Na realidade, temos essa colaboração com Leipzig, que é uma universidade aqui focada completamente na parte de bioinformática, de como nós podemos, né, com essa quantidade enorme, transformar isso numa base que seja, não diria comercial, que seja de fácil acesso, e ela tenha todos os dados que nós achamos importantes para os objetivos fins. Tudo isso tem a ver com esse projeto enorme que eu coordeno, que é um projeto INCT no Brasil. Nós tivemos toda a verba aprovada. A questão aqui não é verba, a questão aqui é saber como nós vamos sintetizar toda essa informação, atendendo os ODS, que hoje, onde nós vamos, temos que estar antenados com todos os 17 objetivos, que tem tudo a ver com o estudo de produtos naturais. E, logicamente, eu achei muito importante ter sido convidada para esse evento, porque eu vejo no Senai, no Sebrae, nessas instituições, uma maneira de não apostar em verba, porque nós temos a verba, mas de nos possibilitar a dar um passo adiante para que, num prazo de 2 anos, no máximo, nós pudéssemos ter toda essa informação de dados químicos, de dados taxonômicos aprovados. Então, na realidade, o meu sonho é ver a certificação dessa base de dados e, logicamente, agradecer a vocês e pedir desculpas, porque na minha agenda estava marcado 13h30. Né? Isso é uma colaboração, nós temos colaborações internacionais que são extremamente importantes, né? E principalmente os financiadores, sem eles nós não poderíamos chegar a lugar nenhum, e os nossos alunos, né? É extremamente importante estar com eles, porque eles, no país e fora do país, são eles que movem nossos laboratórios, todos eles muito talentosos. Eu acho que a maioria hoje ocupando cargos e funções espalhados nas nossas universidades federais, institutos de pesquisa e no exterior, o que é extremamente importante. Muito obrigada pela atenção de vocês e, mais uma vez, mil desculpas.
Muito obrigado, professora. Eu acho que é notável assim, o trabalho que a senhora vem conduzindo nesses últimos anos, né? O trabalho que o NUB vem realizando, né? A construção dessa base de dados, né? A riqueza dessa base de dados e quanto já saiu, né? De produtos, de valor, né? Que eu tenho certeza que é para além do valor econômico, né?
Mais do que é um valor econômico, é muito importante, nós sabemos disso, porque
Né,
isso tem a
tudo.
ver com a nossa economia, com a melhoria da qualidade de vida das pessoas, mas o valor do conhecimento, este fica e pode ajudar as próximas gerações.
Certeza. And now I'll switch to English because we are about to introduce Brian here.
Sorry, Brian. Sorry. Eu esqueci. Mas my slides in English, so okay.
No problem. Uh, there was something in Professor Vanderland's speech, Brian, that mentioned how large amounts of information are now available related to the biodiversity. But the challenge is to transform this raw information into useful knowledge. So I think CAS has all the tools necessary to do that. And with that, I'll give you the word. Thank you very much for your presence here, your availability to be with us. And please go ahead, Brian.
Okay, I hope everyone can hear me. Thank you very much for the introduction. I'd like to say it's a real pleasure to be here with everybody, and thanks for the good segue, Vanderlaan, into this presentation. It is true, CAS does have the tools to make all of this data come alive, and not only do we have the tools, but we also have the expertise, which I'll talk a little bit in my presentation. There we go. Just so you know who you're talking to today, My name is Brian Harkleroad. I am the director of our CIS Custom Services business. I've been with CIS for 23 years. I've been in the Custom Services division, which is a new division. Traditionally, we only worried about tools, or we only worried about products. But now we're working with our clients to come up with custom solutions, and it's a real exciting time to be a part of CIS. My background is in chemistry and computer science, and hopefully you'll see some of that interspersed throughout my presentation today. At the end of this presentation, I'd like you guys to have a better understanding of who CIS is, what we can do, and why biodiversity and finding natural products is so important to us, and how we can potentially help move the science forward. I mentioned I've been at CIS for 23 years. That's because I believe very highly in the mission that not only CIS has, which is, collecting all the information that's been publicly disclosed in the chemical space, but also as part of the American Chemical Society. We are a mission-based organization, and we take that to heart. And you get a lot of really dedicated, smart people that aggregate around this business. And it's pretty exciting to work with this group of guys and gals. And it's great working with customers because they were all very passionate about pushing forward scientific progress. So a little bit about CIS and who we are. We started in 1907, and back in 1907, there's an agreement that there was a problem, and that problem was that there was just too much scientific literature to read. No one person could possibly read all the scientific information that was being published in the United States and around the world. It should seem very, very obvious that someone needed to take care of this. So what happened is a group of folks got together originally in Illinois, and then moving 2 years later into Columbus, Ohio, with the mission of trying to index and abstract the world's information around chemistry. That first year of CIS being a part, we looked at over 12,000 abstracts, and we published 12,000 abstracts. And just a point of comparison, this year we do over 1 million different publications. That's from about 10,000 different journals, 65 different patent offices. And over on the right, you'll see that we have experts that speak over 50 languages. So a lot of the work that we do is in the native language. You might also know CIS from our CIS registry file. That started back in 1965, and it took us all the way to 2009 to create or register 50 million substances. In 2013, we were up to 75 million. 2015, we're at 100 million, and today we're at 220 million small molecules. So as you are very much aware, the amount of science, the amount of molecules that are being discovered is growing exponentially, not at the rate that we can do all of our due diligence in the scientific area to do all the tests we need to do on all these substances. CIS is respected around the world. We support governmental agencies with nearly every major country in the world, thousands of world-class universities and chemical companies, of course. We do a lot of work in polymers and chemicals and, of course, pharmaceuticals. We've recently expanded to get outside of just the chemistry bubble. We're looking at how that interface with life sciences is. We're starting to collect a lot more bioactivity, structure activity relationship data, so we can get people to that next discovery is how can you help the human race and animal kind. So that tells you a little bit of the history of us, but what does CIS do? We are a collection of over 1,000 experts. And what we try to do is we try to take this mess of all this information that's out there. And if you think back to 1907, for instance, people were knowledge poor. They really had to search for the information that they needed. Now we're kind of have information obesity, where we have so much information that's floating around, it's really hard to manage it. And that's where CIS comes in the chemical space, is we can take some of that data, highlight the information that's important, standardize it so it can be searched across different disciplines, and then using our proprietary ontologies, taxonomies we spoke about earlier, and thesauri, we're able to stitch that together so you have a better understanding of what is the history of a molecule or a particular area of science. So we help you create that good vision of what's been done in the past with the ultimate goal of getting you to insights. We provide our products and services through SciFinder, which many of you might have used. If you're on the IP protection side of things, we have STN. And what I think most importantly is we have this new business, which is the CAS Custom Services, where we can help bring to bear all of the people and knowledge that we have at CAS and our tools and our content. We can bring those to bear on your problems to help you get to the insights. So enough about CAS. Let's return to a topic that was brought up by Vonderland earlier, which is the Newbie Project. It's been absolutely amazing working with Vonderland and Marilia on this project. The excitement that comes to this project is immense. So I want to say thank you to them for working with us. It was probably mentioned earlier, but the way that we look at what the purpose of this is about providing a research resource so scientists can access all of these great compounds that have been extracted from the Brazilian rainforest and give it a little bit of visibility to Brazilian scientists as well. So we did start with a very large collection of publications. We narrowed it down to about 5,500 in this first phase. And our goal is to go through that collection of documents and not just pull out every substance, but to pull out the ones that were natural products that were extracted from some piece of flora. So we were able to do that. We also collected some metadata along with it. So not only the substance and the classification of the substance, but the bioactivity information, the source, where it came from, and if possible, to grab the geographic location where that species was being grown. So we saw this as not only a really important project for our clients, we see this as a very important project for all of chemistry. So we were really happy to be about that. Before we go into some preliminary details, let me just remind everyone, because everyone already knows this, why are natural products so important? Why are they important to chemists? And why are they important to the American Chemical Society? If responsibly managed, this is all renewable. And maybe more importantly, it's a perfect model for chemistry. Nature can do things that we cannot do in the lab very readily. And that leads to some very special properties that we would never be able to attain through purely synthetic methodologies. So there's a lot of applications for this. We've talked about many of them as I was listening to Vonderland speak. Fragrances, agrochemicals, feed, cosmetics, polymers, coatings. These are all things that lend themselves to these natural products that we're able to extract from the great biodiversity of Brazil. And it's also important that even if you just look at the plant extracts, this market is very attractive. It's almost $70 billion. It probably will be in a few years. And that's just the small pittance of what we're mostly talking about, or what people naturally think of when you talk about natural products, and that's bioactivity. We're talking about the pharmaceutical industry, which is now at $1.5 trillion a year. So even no matter how you slice it, there's a lot of opportunity in these areas. And I think there's a lot of research to be done and a lot more discoveries to be made. So I mentioned that we started the Newbie Project. We are not quite completed with it. We have looked at and quality assured about 350 documents. This isn't a random 350. We actually looked at the ones that had the most natural product extraction, and we were able to look at metadata to identify those. So we chose the ones that were most rich in the document collection to start on. From just those 350 documents, we were able to extract 1,200 unique natural substances from 280 different plant species. And coming from, that might not seem a lot to you guys. You guys live in the Brazilian biodiverse, maybe the most biodiverse place in the world. I grew up in the Midwest of the United States, where you had two plants. You had corn and you had beans. That's pretty much all we ever saw. So being able to extract natural substances from 280 different species is quite impressive. From those 1,200 natural substances, we were able to identify 100 different biological activities. And what you're looking here on the right-hand side of the screen is really just a heat map of where the substances that we've extracted already, where those come from. So you see some hot spots. I would expect once we get through the entire collection, this heat map, if you will, is going to change a little bit. But we're already seeing some hot spots around Sao Paulo and some other places in the South. It's very interesting to look at this data from a different point of view. And I was thinking to myself when I was listening to Vonderland go over her presentation, I tried to keep my presentation very high level. I thought about digging into the different skeletal and the graphs, the graph similarity that you see across all these molecules. So I'm glad she got a couple slides in there, because if you do look at the scaffolds, it becomes very, very interesting. Not terribly surprising, but I think interesting. I talked about the importance of natural products. We've identified in our collection 165,000 different natural products. It might seem like a lot, and if you compare it to just looking at one biome of Brazil and what we've extracted, and what we extracted already, it's quite large. We found 1,200 so far. We expect that to grow quite significantly. But this idea of having 165,000 natural products, that's a lot of chemicals that we can go and look for bioactivity, look for its ability to have smells or to bind to form biopolymers. There's a lot of chemistry that we can look at in those 165,000 products. But I'm here to say that this is a drop in the bucket. If you compare natural products to all of registry, I talked about earlier, it was 220 small molecules. You'll understand why we think that there's a lot more natural products that can be extracted from the flora in Brazil and from around the world. So we see this as a huge growth opportunity for the markets that operate in these areas, as for us in a business to understand and how do we help people make discoveries. But I think this is just the tip of the iceberg, and we're going to see in the coming years quite a bit of better techniques for extraction, many new compounds that are coming out. So it's a pretty exciting time to be part of this. The other thing that I think could be interesting is when you take the natural products that you have here and you try to project it onto the enormous registry file, you can look at things, not just at other substances that might be biosimilars, but you can also look for how the molecules can be mutated, for lack of a better word, or transformed into something that's a little bit more bioactive. So looking at derivatives of some of these natural products can be quite fruitful. And that's across the board for any type of use, whether you're creating polymers or you're creating something to cure diabetes, for example. So very exciting time. The next thing we did is we pulled out, we looked at the Brazilian documents and we looked at the 1,200 or so substances, and we wanted to look at it from a fairly high level. So we just looked at the bioactivity profile. This is intentionally small, so I hope none of you are out there squinting at your screens trying to find a story here. I wanted to offer this up as a profile of what we have found so far. And if you look at it long enough, you start to see some trends where a lot of the bioactivity explained in the Brazilian documents alone, you see a lot of things that are like defensive in nature. So you see antimicrobial, antibacterial, parasites, larvae, other viruses and fungus, things that have been developed through evolution over time to help the plants survive. So when just looking at these Brazilian documents, you think it's a relatively small niche type of substances that are out there. But if you take these same compounds and you look across all references that's been published since back into the 1700s, you see quite a different picture. So if we were to take those same 1,200 substances and expand it across all of the documents that we have in our collection, a slightly different picture evolves. If you ignore the colors, they mean nothing. But I'll highlight a couple items. You see quite large areas where we're talking about anti-tumor agents. We're looking at things that have to do with the immune system, anti-proliferative agents, immune agents, anti-inflammatory agents. So you start to see a different profile. You look at those substances a little bit differently, and that's where a lot of these discoveries can be made. So I'm going to take just a few minutes to highlight a couple substances and tell you a little bit of the story. This is from a very common plant in Brazil, or so I'm told. Simple extraction, being able to find this coumarin molecule here. And I wanted to highlight this because it's been identified relatively recently in the last 2 decades. And it's also been worked on a lot by Brazilian scientists, which I think is incredibly great. Back in 2003, this was isolated, and there was an initial study as an anti-parasitic. And then a couple of years later, saw analytical determination, and that article was published. And then there were some studies around efficacy and its use as an anti-parasitic. So building on the knowledge about this particular substance over time, a lot of different journals, a lot of different disciplines, and then some use information around topical treatment. This particular molecule, or this class of molecules, is used a lot in ulcers, treatment, skin burns, protection from the sun, and so forth. So to be able to know that you can have a topical treatment that can be an anti-parasitic is quite interesting. And then finally, most recently, in 2020, a lot of work done around SAR data. So that's fantastic. Another quick example, again, from a native plant in Brazil, we're able to extract this molecule, and there's not a lot about it. And that's what's so neat about it. We're talking about 2012. Not a lot of studies have been done on this. And that's why I think the importance of Newbie is so great, is it gives visibility to some of these substances that might only have been studied in South America. In this case, the initial isolation was in 2012. And then in Peru, another work group was working on it. And even though it was originally studied as an anti-herpes treatment, it was also showing some activity with around colon cancer. So it took several years for someone adjacent. I think one of the goals of the Newbie project is to elevate some of these discoveries, and people have a chance around the world to work on them. I'm going to leave you with one extra example of what we've done with other clients. We had a client that was looking for pesticides that were made from natural products. There were a lot of challenges on this project. They had their work cut out for them. There's a lot of data, but the bioactivity data is scattered throughout the literature. They didn't really understand what are the key molecular properties that they needed to look for if they wanted to create a model for predicting that the next pesticide that has all the characteristics that they're looking for. And also, sometimes these properties that are key are not extracted from the original literature. So they came to CAS and asked if we could help. And we were able to help them, try to save them some time in the lab. They did not have the time nor the resources to test every single thing that they could possibly come up with based just on structural moiety. The solution was that we had our subject matter experts consult with them and identify some of the important properties of these substances and some of the scaffold that they share. So we can set up some tests and go through the literature and go through our substance collection to find targets that could be used for training and model. And that is what we did with them. And we were able to reduce the amount of R&D time that went into the discovery of new pesticides. That work continues. And it's just another way of thinking about CAS, not just as a product or a research tool, but helping our clients, like on the Newbie project, meet their business goals. This last slide is an invitation to you to reach out to us anytime that you need some of our insights, but also draw some attention to the fact that we do publish in this area. We publish in journal publications, we write articles, we write reports, and we promote that on our website. If you go to cas.org under Insights and look at some of the other topics. Because of the shortness of this particular part of the talk, I couldn't go into all the incredible things that we're doing with clients, but hopefully this gave you a sense of how we can make an impact in the area of natural products and leveraging the biodiversity that's found in Brazil. Thank you.
Thank you very much, Brian. I think it was very interesting to understand a little bit about the solutions that CAS has to offer. We sure have a time to discuss it more and go in depth in our round table when we will talk about what we saw here today. Now I'm going to call here Yami for a presentation regarding our biodiversity business discovery, our proposal as well. So, Yami.
Oi, tá funcionando. Acho que só falta a apresentação. Apresentação.
Pessoal, pode... ok.
Aí, obrigada. Boa tarde, pessoal, tudo bem? Bom, acho que aqui quase todo mundo me conhece, mas meu nome é Yami, eu sou a pesquisadora da parte de biotecnologia, sou farmacêutica de formação, com doutorado em biologia molecular e celular, e hoje estou trabalhando na parte de biologia sintética. Bom, hoje eu vou apresentar E eu gostaria de começar de uma maneira um pouco diferente. Eu queria fazer uma pergunta para vocês. Só antes de começar, eu queria o pointer. Obrigada. Aí. Aham. Sim, você usa. Obrigada. Então, hoje eu queria começar fazendo uma pergunta para vocês, para saber, entre esses dois itens, se eu tivesse que perguntar para vocês qual desses dois vocês acham que tem o maior valor, se é um Boeing 737 ou uma jararaca da mata, qual que vocês escolheriam? Bom, se você respondeu da direita, se você respondeu o Boeing, eu sinto te informar, mas vocês estão errados. Esse Boeing, ele é avaliado em mais ou menos uns 100 milhões de dólares, enquanto a jararaca da mata, que é nativa do Brasil, produz um veneno, o qual possui nesse veneno uma molécula chamada de bradicinina, que é o princípio ativo do captopril, um dos principais medicamentos utilizados como antihipertensivo no mundo. E isso alcança um mercado de 536,6 milhões de dólares, com um crescimento estimado em 17,3%. Então, sim, a biodiversidade tem valor. Ela garante a nossa saúde, a segurança alimentar, ela é fonte de tratamento para diversas doenças, como o Brian e a professora Vanderlan já falaram, e ela pode modular até metade do PIB mundial. Gostaria de trazer aqui só mais alguns números impactantes. A biodiversidade ela é capaz de impactar até 44 trilhões de dólares. E na indústria farmacêutica, pelo menos 25% dos medicamentos vendidos têm origem em moléculas naturais. E dentro dos tratamentos de câncer disponíveis, pelo menos 70% são de moléculas naturais ou semissintéticas. Para quem não sabe o que é uma molécula semissintética, é basicamente o que a professora Vanderlan e o Brian estavam falando, de usar um scaffold. Então, você usa uma parte de uma molécula natural e, por síntese química, você vai modificando ela. Finalmente, a cada dólar gasto com preservação, a gente tem um retorno de 9 dólares em benefícios econômicos. E eu espero que até aqui eu tenha convencido todo mundo de que a biodiversidade ela é um negócio rentável e sustentável. Na vanguarda disso, em 92, aconteceu aqui na cidade do Rio de Janeiro a Eco 92 e foi assinado o Tratado CDB, que é o Tratado das Convenções sobre Diversidade Biológica, onde mais de 100 países assinaram esse tratado com o objetivo de conservação da diversidade, o uso sustentável, a repartição justa e equitativa dos benefícios e acesso ao patrimônio genético. E, como não poderia faltar, o Brasil sempre teve um papel de destaque, né, nesses tratados, nessas atividades. E não é por menos, tendo em vista que nós temos 20% da biodiversidade do mundo, dividido em 6 biomas que são incrivelmente diversos também. Nós temos a maior floresta tropical do mundo, 70% das espécies de animais e plantas estão aqui no Brasil, e, em média, anualmente são descobertas 700 novas espécies de plantas ou animais. Ou seja, nós temos uma riqueza imensa, a qual a gente realmente deve explorar e saber utilizar. Entretanto, a gente tem subaproveitado esse potencial, mas graças a pesquisadores como a professora Vanderlan e outros grupos de pesquisa, estão incansáveis aí correndo atrás de recursos para conseguir identificar essas moléculas e explorar comercialmente para que tenha um retorno econômico. Alguns exemplos de moléculas identificadas no Brasil para o setor farmacêutico são a bradicinina, que eu já falei, a pilocarpina também é um bom exemplo. A pilocarpina ela tem sido utilizada na forma de colírio para redução de glaucoma. O lapachol é uma outra molécula proveniente do ipê roxo, tem atividade antimalárica, anti-inflamatória, e também alguns estudos demonstram atividade antitumoral. Além disso, o Asteflan, que a professora Vanderlan falou, é o primeiro anti-inflamatório 100% produzido no Brasil, com extrato da Cordia vernácea. E, enfim, nós temos que nos orgulhar muito dessas conquistas, porém a gente sabe que estamos subaproveitando o nosso potencial. Bom, então quer dizer que nós temos a maior diversidade do mundo, milhares e bilhares de moléculas e metabólitos secundários, peptídeos, proteínas que podem ser bioativas, e nós temos que achar utilidade para essas moléculas. Bom, atualmente, a nossa capacidade de identificar e caracterizar essas moléculas e proteínas aumentou muito o nosso poder de identificação, com as análises de metabolômica, genômica, proteômica e transcriptômica. Entretanto, apenas identificar e caracterizar e publicar, ou mesmo só patentear essas moléculas, não vai nos trazer um retorno financeiro. O que nós precisamos fazer é encontrar utilidade para elas e aplicação na indústria, seja ela farmacêutica, cosmética, agronegócios, e é por isso que o ISI Biosintéticos e Fibras lança um novo modelo de negócios chamado de Biodiversity Business Discovery, onde a ideia é pegar essas moléculas, essas coleções de organismos, plantas, essas informações, triar dentro delas moléculas que tenham atividade que nos interesse e que a gente consiga direcionar para o setor agrícola, para o setor de alimentos e bebidas, para farmacêuticas ou HPPC. Bom, mas e como que a gente vai fazer isso? Então, o nosso modelo que estamos propondo é um pipeline de descoberta de moléculas. E como que ele funciona? A ideia é que a gente consiga triar o maior número possível de moléculas, ou, enfim, peptídeos, proteínas, para que a gente consiga avaliar frente a um alvo desejado. De uma maneira simplificada, nós podemos partir desses bancos de dados, onde nós teríamos essas moléculas disponíveis, e realizar triagens em sílico, que basicamente quer dizer que a gente vai fazer análises computacionais. E para isso, o ISI Biosintéticos e Fibras conta com uma equipe especializada em bioinformática e também outros parceiros que possuem equipamentos que dão processividade computacional para esse tipo de análise. Nessas análises, nós conseguimos predizer interações entre proteína-proteína ou molécula-proteína, sempre buscando o nosso objetivo final. A ideia é que a gente consiga levar para a bancada ou para a parte experimental apenas as moléculas mais assertivas, ou que a gente consiga sair de um cenário onde nós temos milhares e milhares de moléculas e a gente consiga reduzir esse cenário para que seja factível a validação em bancada. Bom, mesmo assim, às vezes a gente chega na bancada e ainda assim tem muitas amostras para fazer a triagem, muitas amostras para trabalhar. E nesse sentido, o ISI Biosintéticos e Fibras conta com uma estrutura completa de automação. Automação nada mais são do que robôs que, quando programados, repetem atividades. Nós contamos 2 Hamiltons, que são robôs pipetadores, também temos Opentrons, alguns equipamentos como colony picking e dispensadores de pequenos volumes. A ideia é que os protocolos sejam desenhados de maneira simples e que os resultados sejam de fácil leitura, onde nós conseguiríamos responder se, por exemplo, uma molécula tem atividade surfactante. Então, os robôs conseguem pegar através de uma biblioteca de compostos naturais, por exemplo, e avaliar se, frente a um alvo ou a sua atividade que está sendo medida, a gente consegue identificar a resposta que gostaríamos de identificar. Bom, aqui eu trago um exemplo, né, de como poderia acontecer esse fluxo. A gente pode iniciar a nossa busca através de um extrato de planta, com suas frações, ou de uma coleção de micro-organismos retirada do mar, ou ainda de bases de dados, como a base de dados apresentada, a NUB. Através disso, a gente tem que selecionar o nosso alvo. O que estamos buscando? Estamos buscando um surfactante ou estamos buscando um tratamento para um determinado tipo de câncer? A partir disso, nós vamos desenhar a nossa ideia experimental. Podemos realizar uma triagem em sílico, digamos que a gente esteja procurando uma proteína que ligue em um receptor específico. Nessa triagem em sílico, realizaríamos a interação entre as moléculas e o nosso receptor de interesse. Finalmente, após isso, poderíamos levar ou não essas análises para a bancada, sendo validadas via HTS ou experimentos convencionais. Aqui eu trago só um exemplo, né, da plataforma da professora Vanderlan, que é a Nube, a qual ela já demonstrou, mas é muito interessante porque nessa plataforma nós conseguimos associar a estrutura da molécula à sua espécie e onde ela foi identificada, assim como o trabalho científico publicado. Também é interessante porque nós conseguimos pegar as informações dessa molécula e utilizar nos nossos programas computacionais para realizar essas modelagens ou simulações de interação. Então, aqui nós temos 2 exemplos de fluxo. O primeiro, nós iniciaríamos com a triagem das moléculas de um banco de dados computacional, realizaríamos essas triagens em sílico. Nós podemos tanto validar posteriormente em bancada, quanto iniciar diretamente os testes em bancada, se a gente já tem algum indício de quais moléculas gostaríamos de analisar. E, finalmente, nós teríamos como resultado as moléculas mais promissoras, que na indústria farmacêutica se chamam os hit leads, os quais nós investiríamos mais tempo para buscar a sua atividade e validar, porque os ensaios de HTS, eles são apenas um indício. Posteriormente, nós teríamos que validar e fazer outros ensaios para verificar se sua atividade realmente é um verdadeiro positivo para aquela resposta. Entretanto, o ISI Biosintéticos e Fibras entende que nada disso vai acontecer sozinho. Nós contamos aí com uma rede de cooperação, onde nós entendemos que precisamos interagir com diferentes bases de dados, pesquisadores e iniciativas também do governo federal. Um bom exemplo é esse SI-BBR, que é o Sistema de Informação da Biodiversidade Brasileira, que também está associado ao GBIF, que é uma base de dados que também contempla a biodiversidade mundial. Então, eles estão diretamente em contato com o GBIF. Também chama atenção aqui para o SEMBAN e para o PPBio, que estão um pouco mais focados na biodiversidade amazônica, e para o Instituto de Desenvolvimento Sustentável, Mamirauá. Esse aqui é um instituto que ele é idealizado também pelo MCTI, e ele fica lá na Amazônia. Ele possui diversas unidades chamadas de laboratórios salas, que podem ser alugados, e a gente pode também começar uma interação com eles para ter acesso a locais da Amazônia que são menos acessíveis. Esses laboratórios, então, podem fazer coleta e análises de plantas, espécimes, fungos, bactérias da biodiversidade amazônica. Então, eles estão sempre abertos para contato e eu incentivo todo mundo a ter uma conversa com eles. Bom, aqui eu trago, né, a página do SI-BBR, caso alguém não conheça. Ela é uma página muito interativa e interessante, e todos nós podemos depositar os dados que estão sendo criados, tanto os dados genômicos, quanto catálogos, taxonomia e até geolocalização. É interessante porque você consegue fazer listas e filtrar as espécies que você está procurando, e também filtrar via geolocalização, né, no mapa. Você também consegue contato dos pesquisadores que identificaram e das coleções de organismos ou coleções de animais que você poderia acessar. Nesse contexto, então, o BBD, ou Biodiversity Business Discovery, entende que nós devemos trabalhar em grupos, né, e em redes, no formato de rede, onde nós ficamos apoiados nessas iniciativas e também com pesquisadores como a Vanderlan, para encontrar, então, utilidade e aplicação dessas moléculas que são provenientes da biodiversidade brasileira, para finalmente a gente encontrar atividades como agentes biocidas, novos fármacos, flavors, fragrâncias, surfactantes, entre outros. E dentro dessa ideia, né, de descobrir, de encontrar e de criar utilidade, eu trago aqui uma frase do biólogo Thomas Lovejoy, que já no passado dizia isso, e eu acho que isso é muito atual e presente, mas é importante que a gente realmente aplique, que uma floresta ela é como uma biblioteca de livros que não estão sendo lidos, e explorar a sabedoria contida nessas bibliotecas pode nos levar à descoberta de produtos inovadores e novos mercados. E eu diria até mercados que ainda nem existem. Então, é isso. Eu gostaria também de divulgar a nossa nota técnica que foi publicada hoje. A gente tem aqui um QR Code, caso vocês queiram acessar. Ela tem a versão em inglês e em português, onde a gente fala um pouquinho sobre essas metodologias, né? Eu gostaria também de ressaltar que, tanto para as metodologias computacionais quanto para as metodologias HTS, já existem estudos publicados, kits prontos, mas nós também podemos desenhar um modelo experimental o qual possa atender a indústria ou a demanda que está sendo pedida, né? Então, é isso. Lá vocês vão encontrar mais detalhes e a gente fica à disposição para conversa. Muito obrigada.
Muito obrigado, Yamin. Quer dizer, essa é uma iniciativa relativamente nova, né? Mas que a gente vê bastante potencial, né, de desenvolvimento, sobretudo trabalhando em cooperação. Acho que esse é um ponto fundamental quando a gente fala da exploração da biodiversidade. Em relação ao professor Renato Luciano, infelizmente ele não vai conseguir estar conosco hoje, teve, né, algumas questões. Então, nesse momento, a gente, né, tá indo para o nosso coffee break aqui presencial. Às 3h20 a gente retorna, então, para já começar a nossa mesa redonda com os nossos convidados. So, Professor Vanderland Bryan will be returning at 3:20 to start our round table here, where we can discuss what we have talked today. And so I'll see you in a little bit. So bye-bye.
Know the chemical you can see that is compound that you know very well. So, and nowadays I think most of the international multinational companies doesn't have too much interest to discover new skeleton from natural products. However, they have a lot of interest into to study these classes and to modify or to improve the activities or pharmacological, or cosmetic, or supplement activity of these compounds. You have a lot of compounds. But in Brazil, we have 6 biomes, and you don't have too much studies on the biodiversity. But you know the class of compounds. Chemosystematic is also a very nice approach. I was in Germany recently, and we discussed a lot about the focus on chemosystematic to find new prototypes.
I was curious about this approach. I do not know it. Can you explain a little bit, Professor? Chemosystematics? You are muted.
Chemosystematics is a very nice scientific approach that was settled by Professor Gottlieb. He was very famous. That we suppose, if you know all classes of secondary metabolites already isolated, spread out, and you have idea about the biosynthetic pathway, you can use some very simple mathematics to calculate the level of this skeleton can modify, and also the oxidation in natural products. And so you can do phylogenetic pathways of species associated with class of compounds. When you have this idea that you have, my colleague here, she mentioned GNPS, that is a very important database in the United States from Peter Dorenstein. If you have this idea all organized, you don't need to spend a lot of money, use HPLC, NMR, that is very expensive in order to study species. If you know the species, you know the chemical composition of this taxon, you are be able to predict compounds. Okay, you can find one compound that is not in that biosynthetic pathway, but it's very rare. It's not so common. Most of the nature produce secondary metabolite based on precursors of photosynthetic pathway. That is very important. This is very nice nowadays to have this approach to looking for new compounds. I have a student, she did a PhD with myself, and now she's in a postdoc in San Diego, work on Peter Dorenstein, and they have this approach. It's very nice. It's very nice that you combine genomics, chemistry, and chemosystematics.
Nice.
Very nice, Professora Vanderla. And it is a very useful tool to investigate the metabolomics, or you can also use the transcriptomics data, so you can predict what kind of metabolites based on the synthetic pathways, the enzymes, and so on. But also we have to be careful, even though it's a very powerful tool, we have to be careful because it's just a prediction. We don't know if that pathway is activated on that organism or not, but it's a very good tool, and it also depends on the organism you're working with. I mean, if you're talking about metabolites of bacteria, it's easier to work with meanwhile, if you're talking about plants, it's more complicated. So you have to be a very well-trained bioinformat, but it is a very, very, very important tool that can save us a lot of time. But concerning your question, what was it? Like, how can we start?
How can we start with?
Okay, so I think the most important thing is to be very clear about your question. Like, what are you looking for? You know, like when we look for pharmaceutical industries, we have the first antibiotics like penicillin. So we have like the strategy of discovering it by serendipity. So you can, you know, accidentally find an effect or looking for its phenotype. But nowadays we have a very sophisticated tool to, you know, identify the mechanism of action. So you can do a target-based approach to identify which kind of compounds or which kind of proteins or molecules can induce a response on one receptor, for instance. So I think nowadays, the most important thing you have to ask yourself is, what am I looking for? Am I looking for an antibiotic? What is the mechanism of action I'm expecting? So you go for that question and you design all the experiment looking for a molecule that activate your receptor. So the most important thing is know what you're looking for, make your first question and design your experiments. And it has to be a very clear and simple question, so you can screen a lot of molecules, you know. So I think for me, this is the most important thing right now.
Great.
Yeah.
So I have a slightly different perspective coming from a chemical informatics background. The two methodologies that were just explained are very, very reasonable. So I'll take an antagonistic approach. I mean, there's the other idea of finding substances that have been classically been used as medications or have some express themselves in some way. and try to isolate as many compounds as you possibly could, with the hopes of being able to isolate them, look for commonality, and from that be able to determine other possible pathways or to validate particular pathways. So just I always think it's very interesting to see the new compounds that are being isolated to understand what their function is in the plant, but also to find out what other mechanisms are available to you from a pure chemistry point of view. Because as I said earlier, most of the time you would have no chance of being able to synthesize that, nor would you even think about trying to synthesize something that you might find in an isolate or something like that. So almost like a backwards view that was discussed prior.
Thank you, Brian. The other question I have, I think all you have been working for many years now with biodiversity and other types of developments and discovery. Could you take us maybe through a little bit about the development of a specific molecule, of the identification of specific targets that were successful for you, so we can have more perhaps practical examples of things that we can do, how we can approach some challenge on the discovery? Maybe Professor Vanderlaan or Brian?
Brian, maybe Brian. I am a scientist in the lab. I think I have idea of some targets, but I don't know. It's not easy to identify what some company or some program are interested. If you have, because when you talk about the Brazilian biodiversity, is a lot of information, and also some information could be start all step again, because I don't believe, because we have a very difference from Chinese traditional medicine than Brazilian traditional medicine. Because in Brazil, you have a lot of information that is from the antecessors, Indians, and etc. But this information is not so organized. You study several plants that supposed to be compounds with determined activity. When you isolate the compound and you have the pure compound, they don't have any pharmacological cosmetic properties. This is also a question that when you work on natural products, you need to have attention to this focus, because there are one big difference when you, okay, I use metabolomics, I use omics, because I have focus in some classes, but from several information about Brazil, you don't have too much biosynthetic pathways, because sometimes I think my colleagues confuse the term. Biosynthesis is to study all step that one organism biodiversity use to synthesize some compounds. That's completely different when you go to the lab, use molecular biology, use all chemistry, all things. It's completely different because we don't know all enzymatic systems that are responsible in nature for some transformation. This is wonderful subject, but sometimes you don't know anything about the plants of equatorial. You have several studies in the United States, in Europe, but with plants of this temperate zone. That's completely different from the biomes that you have in Brazil, mainly equatorial. That's different from Atlantic forest. That's tropical and equatorial.
Yeah, maybe let me reformulate. The question is more like, I know Brian is helping a lot, Professor Vanderlaan, for example, with the screening of molecules. Can you go let us guide us through the how you're doing that specifically? I think Brian didn't have quite the chance to get in details in his presentation, but I think now it's a interesting time for us to do that.
Yes, and I would like to know, Brian. It's very important for us.
Yes. So, when we started this project, we went down some dead ends. Like, what was the most efficient, the most, not just the most efficient, but what was the most, what was the best approach to make it fairly efficient, cost effective, speed to delivery. We had a lot of different success criteria when we went into this. So we looked at using AI algorithms that we've developed within our organization to predict certain things. What is the proximity of a bioactivity to a particular substance? Being able to utilize things like optical structure recognition is another thing that we looked at to try to automate as best we can to tie together bioactivity, genus, and location. So we attacked it with algorithms first, and we spent a lot of time invested in this, really because we wanted to learn. This is an area that we're very interested in. As everything becomes more automated, new technologies around natural language processing came on board with large language models, which we are training with our data. So we had a lot of technology that we could bring to bear on this. We ran through it, and we did some tests with running pure algorithms, then doing some auditing with human beings and so forth. What we ended up doing is the tried and true, take a chemist, put them in a room, give them the documents, and give them the tools that they can extract it. That is always going to be our gold standard. We had hoped to shave some time off of it, but when we knew how important this project was and the high level of fidelity that was needed, we decided to fall back on our tried and true method of having chemists read through the document, hand extracting the substances, and making determinations of whether it's a semi-synthesis or it's a full natural product, a little bit of information about how it was extracted when that's available, and then, of course, tying it to a plant or an animal, along with the bioactivity. That was all done manually. At this point, we still haven't gotten to the panacea of having an algorithm to pull all that information with very high confidence. So what could have turned into a sexy story about how technology came and saved the day, it turned out to be good old know-how by chemists and the ingenuity of humans to be able to tie those things together.
I'm really impressed that you did manually. That seems a lot of work.
It is a lot of work, and we do have processes in place that we use for our normal what we call curation.
Yes.
So I think I mentioned it earlier, but we go through 1 million documents a year. So we have several hundred PhD chemists that are trained and with tools to be able to extract this large amount of information so quickly. But thank you. It's quite a testament to the dedication that we have with our scientists here.
Yeah, and this is so important. I'm really happy to have the contact with Professor van der Laan and Marilia as well, and to know how important it is to have all the documents, like the system that I mentioned, the SIBBR. That's the National System for Biodiversity. They have also been collecting data from books, so like we have a lot of catalogs, taxonomy, and it's a hand work that they have been doing as well to get all the data and all the information available for the whole world, so I know that's what you guys have been doing for a long time, but in Brazil, I think we are starting to organize out the information. So, wow. But it's very nice to know that you guys had a lot of work, and now it's getting easier with the AI. But I know we still have manual and researchers working on that. But it's very impressive.
Yeah. Sorry, Brian, maybe I think very challenging for us still have been to extract material the extraction of material, because so long in the past you used several kind of solvent that nowadays is not useful anymore. You need to respect the ODS to 2030, the protect to the environment, the protect ourselves.
Mhm.
And this is a very challenging nowadays when you need to isolate several compounds. We have the in our lab now, we are green. You don't use chloroform, you don't use chlorates, but it's a challenge because sometimes in the past we use some kind of solvents that you can isolate so many things. You can have a idea about several classes of secondary metabolites that you have in a leaf, in the fruits. But when you have selection of the extracts, sometimes you lose. So do you have an idea about the work that have been published nowadays about these questions related to isolate echoes, to extracting echoes or ethanol? Don't use methanol, that is very toxic, and chloroform and something like this. That is very important when you focus on isolation of pure compounds. If you work in a mixture, no problem. But when you focus, it's
Yeah,
very...
so if I can generalize a little bit, it's kind of in the area of green chemistry. The American Chemical Society has been at the forefront of promoting green chemistry, and CIS, of course, is dedicated to that as well. We have been working with different organizations, mostly in the commercial space, that have a particular solvent or a catalyst maybe where it is not quite as green or is forbidden now, and they need to be able to eliminate certain solvents. Let's use solvents as an example. They no longer want to expose their workers or the environment to those kinds of things. So some companies have engaged with us to try to find some alternate solvents where we can go back and look at a lot of the different transformations that we've captured over the years in our reaction collection. So even if we don't have the reaction that they're performing at scale, we can make certain assumptions and give them some really good ideas and a consulting approach about another slightly tweaked process, but with a different solvent, whether we're increasing heat or we're doing something else, to try to find substances that are used as solvents that aren't as risky. So we collect a lot of safety information, GHS codes, having talked about handling, all of the regulations around handling and mixing and storing and all that stuff. We can take all that information into account to try to find alternate solvents, and sometimes we can help them find alternate reagents and catalysts as well. It's a big focus right now, as you guys know, to get rid of some of the very hazardous things that take place at an industrial scale, because they have huge impacts on the environment. And looking at data is one way of getting there. You don't always have to go back to the lab and try to work this out. You can solve yourself a lot of lab time by looking through the data that's already been disclosed in the literature, and we can certainly help. It doesn't hurt to spend time in the library sometimes.
Thank you. It's very important to see how you can prepare samples. It's fundamental, and there's a challenge nowadays. If you have so many sophistication.
Yeah.
Yeah, thank you. I was going to ask what is the advantage of using a nature source library of molecules in comparison to a synthetic library? Can you explain a little bit?
Yeah, I think before I answer the question, I just want to contribute with the question of Vanderla, the former question. I mean, usually companies are going for greener paths and greener synthetic pathways to get to the compounds, but I'm going to be cheering for the biotech here, that sometimes you can try to replace all the chemical synthesis with a biosynthetic pathway. So you can, you can develop a more sustainable biosynthesis and then replace extraction from a plant. But of course, we're talking about industrial scale and a lot of R&D. So it really depends on what you're working with. But we have been advancing a lot with biotech tools and technologies. So I'm pretty sure that in the future, these kind of developments and genomic modifications will be so much easier as it is now, like CRISPR and stuff. So I see a greener future and a easier way to replace a synthetic pathway. But anyway, about your question, what is the advantage? Well, I think both Brian and Vanderlei has already answered this question, but it is very nice that when we talk about libraries, like compound libraries, in some years ago, people stopped looking for new drugs in natural compounds because it is hard to work with plants. It is not easy to extract the metabolites. It's not easy to work with fractions. It is not easy. It's easier to work with, already prepared libraries and stuff. But we, like the pharmaceutical industry, kept on rediscovering the same molecules. So they realized that we have to go back to the natural and we have to have diversity. So nowadays there are a lot of different kind of tools that you can use to quantify what's the quality of that library of compounds. And usually a natural library has a lot of chemical diversity. But a chemical library, just a chemical library, does not go to the other way. It's not as diverse as a biological library. So biological libraries has a lot of chemical diversity. So we have a lot of information in there that we can use. And also when we're talking about pharmaceutical industry, I'm really sorry to bring pharmaceutical because I'm a pharmacist, so this is my my point of view. But anyway, when you're talking about evolution, the whole nature has spent a lot of time dealing with viruses and bacteria and parasites. So they have a natural mechanisms to deal with the challenges. So the logical path, the logical thought, that thinking we should have is mimic the nature, mimic their metabolites, because they are actually working for millions of years. So this is the advantage. They have the deep learning, like an AI. So it's a very sophisticated AI for millions and millions of years to face viruses, bacteria, parasites, and everything. So this is the most important advantage for me.
Do you guys want to comment? Brian, Professor Vanderlei, please.
The only thing I'd add is that I thought you put it very eloquently. The fact that nature has been working on some of these problems for so long, we are kind of newbies in this area, especially when it comes to the chirality and the stereochemistry that I mentioned earlier. Knowing that it is bioactive already, when you're working with a database of naturally occurring products, it's there for a reason. Nature usually doesn't create things that have no purpose. So suppose you just take the registry for example, it's got a lot of substances, a lot of them have no bioactivity whatsoever. So you're weeding through a lot of things that have nothing to do with your area. Even though they might structurally, you might get some you might have some idea that it could be bioactive. It usually isn't. It just isn't. But when you're dealing with natural products, your probability is much, much higher.
Yeah, thank you. My other question, when Brian made his presentation, he showed the charts with the different properties of the database you are screening. And I think when you look at the biodiversity, many times pharmaceutical related properties stand out, especially because of the volumes we can extract many times from plants and so on. But do you see other types of molecules, properties that stand out in your research, or that you came along the years? I mean, agro-industrial properties and so on are you also mentioned, but are there others that we can talk about?
Well, the newbie data that we've been working with is really focused on the bioactivity stuff, but we all know that there's a lot of other applications for natural products. So there's other collections that we have created over time where it's dealing with biofilms or any number of things, whether they're going to in a fragrance or they're going into new building materials. There's so many different uses for it. So the newbie data was focused on one thing. What's great about my job and my colleagues' job is we get to work on very diverse sets of projects. And it's amazing on how many clients come to us and want to talk to us about the green chemistry and minimizing their carbon footprint, using natural materials. So that's industry finally catching up with the rest of science to say there's a lot of good chemistry that's in natural products. So there's so many different uses. Just looking at newbie is kind of focused on how can it be used to treat the human condition or animals. There's just so many different applications. There's people on this call probably have worked in these areas where they've been able to take advantage of all the other the chemistry that's involved in natural products. Certainly, there's a lot of stuff out there. When I was looking through the data very preliminarily, I was amazed by how many solvents were in there, other things that participate in reactions, which you don't normally think about. I didn't have a chance to go back and look at them, but I can guarantee you I will at some point when this project gets near the end. It's going to be really exciting to see the different uses that we find, even in the answer set that's focused on bioactivity.
Professor Van der Laan, do you want to comment?
I agree with Brian. Most of work that we have done, we have analyzed in the literature, are focused on, I think, looking for some activity that is so long time. You see that nature produce. It's amazing because we're looking for in nature compounds that can be useful for us. However, the development of the earth, all organisms in earth, didn't produce anything for us humans. Plants, insects, animals, and all that are formed by a huge biodiversity usually use this fantastic chemical library for defense, for regulation, for many things in the nature, not produced for us. So it's fascinating because you use this for us. Focus on activity, I think it's the most common that you see, because when you work, I work a lot with companies. They don't interest in the compounds. I would like to see if this compound is nice. You're looking for. One very old example of our studies in this country is related to jasmonic acid. It's very common compound in several kinds of plants. Very usual, nobody. I don't have interest because I'm a science. I needed to looking for something that I can publish. So if you see in Lancôme and some several of very important companies, jasmonic acid is a principle of a very, very, very sophisticated cosmetic that nowadays. in the past, when I was in the University of São Paulo, we take this compound and we put in the trash because you don't have interest. It's non-compound. So, because we're looking for the activity, sometimes in the academics, you need to have a new compound, new structure, and nowadays it's not easy. So this is one, I think, is one reason that companies looking for compounds that you have the structure and you can improve the structure. This is very important because when you, so many, I think, and we talk about biosynthesis, it's a very complex text to study biosynthesis from plants that you don't have idea. If you have all sophisticated biotechnology, sometimes biotechnology doesn't work to study biosynthesis pathways to discover step for step what the origin and produce this in lab. It's not easy text until nowadays in the United States, in Germany, in everywhere. So this is one, I think, reason that nowadays natural products is not so in the top for the companies when as 10 years ago. It's important, but you need to focus on some kind of that the companies are interested. I don't know. It's very complex, and sometimes I am so.
Yes,
yeah.
it is hard sometimes to do the biosynthesis, but there's a lot of companies like Camiris, they have like these strains that can produce, like, for instance, terpenoids. So you can use this terpenoids for lots of industrial applications. So from farnesin, they can get the molecule to, you know, flavors or I don't know, how do odores, fragrances, and so on. But it is very important to note that sometimes you invest a lot of money, like building a strain that produces a lot of farnesin, but then you actually save a lot of money because in the end you can, you know, you can do this semi-synthetic pathway. You start as a biosynthesis and then you have one molecule that you can, you know, drive it to different industrial applications. So it is actually a very, it is a smart investment. So it is a lot of money, a lot of R&D, but also when you have an interesting molecule or an interesting scaffold, you can drive it towards where you want. So yeah.
Great. My other question regards how can we scale up the business that we find in the biodiverse. So we go through a library, we found some opportunities. How can we, from a plant or a microorganism, think about scaling up the production and so on?
It is a very challenging for us to scale up, because when you work in the university, is a lab that you have a fantastic lab, but it's academic. So we, in my presentation, I put Senai spectables with a cross that we have patent. We have companies interested in that. Is piperidine, is a alkyl long chain piperidine alkaloids. It's fantastic, not only for the knowledge in natural products, but the activity and some company. But how you can synthesize this, because nature doesn't produce enough material to continue the step of preclinical 1, 2, clinical. So this abandoned. There are so many compounds, even in big companies. I work with big companies. There are so many compounds, very important, that's abandoned because it's almost impossible to scale up, because one cash is scale up to make all step of the studies in chemistry, in pharmacology, and toxicology. The second is to produce in scale to go to the market and to still with the same properties. This is a challenge nowadays with natural products that you know that is a new compound. So I can understand why companies nowadays focus on the compound that you have so many ideas about the structure, about the classes. So I think it's easier to study, or the biotechnology sometimes doesn't work, or who knows, domestication of plants that should be nice. You see that in Malaysia and also in Africa, but it's not easy for us in Brazil. You don't have so much company that can domesticate plants here.
About scaling up, I think it's hard.
Yeah, maybe by technological process.
Yes. I think it's hard to scale up when you're talking about plants, of course, because extraction.
Yes, plants, because microorganisms doesn't have problems.
No, it does have problems. Of course, there's a lot of R&D, but I think that once you can, build a strain that it's stable, and of course, there's a lot of R&D and a lot of research around it. But once you have managed to produce the compound you want, and do all the downstream as well, it is a very successful to scale up, so it is possible. I was commenting with the girls about the opioids from the papole. I don't know how to say in English, papola. Yes, there's a paper in the US where they have modified an yeast to produce some precursors. So it was a very hard work because they have to modify a lot of enzymes in the yeast path to get to the precursor. But it was very, very interesting how you can, modify a whole the whole metabolite of that yeast to get to the precursor and not needing the papole. So I mean, it is possible. That's a lot of R&D, and as Professor Vanderla said, it's a lot of investment, and this is why usually pharmaceutical companies has the potential, and the money to invest on it, because you have the feedback and you have the return. But nowadays, and also there's other precursors that are natural, for instance, in fungal cells, that you don't need to do genetic modification. You can just adapt the strain, like evolutionary pressure to this strain to produce more of what you want. So it depends on the compound you're talking about or the molecule, but there's always a way, and I think biotech is the future way. And once we can move forward with the technologies, and once we can transform the cells with the less money, and when we know in the future, I think the technology will be cheaper, so it will be easier to do R&D doing by biotech.
Great. We are approaching the end of our event here. I'd like to thank so much all of you for the presence and all the great insights you gave. So we can end here. I would like to. I was thinking that for me, biodiversity research and so on is collaborative in nature. I think we are here together. It's a perfect example of that. So maybe I would like to have a few comments of each one of you regarding how can we collaborate with each other to make research, to discover new molecules and so on. How can we gather other stakeholders like government, funding agents, companies, and I don't know, to work and find these new opportunities. So maybe Brian.
Sure, I'll start. Since we're ending soon, I would like to say thank you for holding this discussion in English. My Portuguese is non-existent, so thank you for doing that. It's very kind and courteous. How do we work closer together? Our organization has always been about forwarding science and being collaborative. Not always do we look outside of our own borders. We are the American Chemical Society, but we are very much a global organization. So we very much appreciate collaboration. I think that has been shown with our work with Professor Vanderlyn and her team. We love these kinds of projects because we get to do something important, and that is advancing science and working on some of these projects. So how do we foster it? I think is just reaching out and asking questions. Asking questions doesn't cost anybody anything, but it builds a relationship that can pay dividends in the short term and in the long term. So whether you're reaching out to CIS or big pharma, or it never hurts at all to ask for help and to ask questions, because you'll always at least create a relationship. And whether something can work out or not is kind of secondary. So I'd say always keep that idea that people are willing to help, and we all have the best goals in mind, which is forwarding humanity's struggle through our lives.
Thank you, Professor Vanderlyn.
Okay, thank you very much for this invitation. It's so nice to discuss about natural products, biodiversity in Brazil. And also, I would like also to thanks to CAS. We have a strong collaboration nowadays because we have this collaborative work. In my mind, in my dream, should be to have a natural product database. This is the focus that I try to show you. We advance a lot, but it's a very hard test because when you evaluate that you have around 50,000 compounds isolated, distributed in so journals to organize this in a certified database is a challenging. So, but I didn't talk today, but I saw nowadays with a very important focus to study compounds from our biodiversity is that I changed it, is forget a little the small molecule. And now I try to work with peptides, orbitides that is new ones, and big peptides. And we have several recent reviews in Nature, in Science, and very good journals about the importance of peptides for us. And you don't have almost any compounds of this class is isolated from Brazilian biodiversity. So it's a very important field nowadays. If you see the biodiversity as a source for bioproducts. Thank you very much for this. I will plan to go to Rio to discuss with you about the NUB database and so many things about this, because I would like to stay with you today, but the price of the flight is impossible. Almost 6,000 reals to take a flight for 45 minutes. It's amazing. But I plan to go to you and to study, to discuss with you. That is very important, our database. But I think the focus on natural products nowadays in Brazil should be looking for peptides. You have several peptides in clinical phase now. You have several good articles about this, and is a very, I think, very important field for science and technology in Brazil related to biodiversity.
Very nice, Professor. We are looking forward to collaborate with you. Amy?
Thank you so much. I'm really glad to share this afternoon with Professor van der Laan and Brian. I have learned so much with you. Thank you. And thank you for the invitation. I think that for me, the strategy we should do is we should collaborate, of course, but we should also claim for the government initiatives. We need the government support for for the researchers, and we need the country to value our work. So once we have, we feel like we are valorized, we have a spotlight in the society, I think the easiest it is to move forward with the bioeconomy. So I think we should collaborate, but we should also focus on education from child to, university education. So for me, this is the path. We need to ask and we need to demand the government support. So this is why I think I have been looking for the MCTI contacts, but because we cannot do anything alone. So let's, add our strength, the Brazilian government and the international initiatives like the biodiversities, because also the other countries should be very interested in what is in Brazil, because we have a very wide territory, a lot of molecules, and a lot of diversities that can be a global, source for health, food, and so on. So government and collaboration, that's it.
Okay, thank you very much. I mean, so now we are ending our webinar here. Once again, thank you very much, Brian, Professor van der Laan, for your presence, your great insights. Just to reinforce once more, we are now releasing the technical note regarding the biodiversity business discovered. It's available at Senai website, so you can look it up. Amy also shared the QR code in the beginning of her and at the end of her presentation.
Yeah.
And it's available both in Portuguese and in English, so
yes,
it's accessible for all. So thank you very much and see you next time. Bye-bye.
Thank you, guys.
Thank you.
Thank
Bye.
you. Okay, bye-bye.
Bye-bye.
Thank
Talk
you.
soon.
Bye-bye.